Hello, people, people, people who need people. Oh, that's bad. That is really bad, Dr. Slush. Not do that anymore. Okay. What do we got? 14 people? More on the way, I hope. It's a little bit early. So, did you guys, let's see your faces, everybody. Michelle Simmons, Asha Gupta, Vanessa G, Rocky Raccoon. Okay, where are we now? There's Jordan Fine. Fine man. Fine lad, that boy. Sammy Robinson connecting. Okay, any more faces out there that you want to share with me? Estevez, Turner, Jordan Fine. There's Estevez. Bing boom, bing boom. I'm waiting until we get a bunch of people because I want to ask you guys a question before we get started. Camera hasn't been working for some reason. Okay, Lee, we'll let you go today. Malak, 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 Malak. Malaka. Abdu Sabor showing up. Yay, Abdu. I gotta say, I've said this before, but as I look over the crowd, it's like there aren't a lot of white guys out there. It's such a diverse group in biology in Plattsburgh. I think that's a really cool thing. Got Jordan Fine, our token white guy. Sammy Robinson. Okay, well, we got, what do we got? 24, 25 out of about 30. Let's wait till Megan connects. Okay, so you guys probably saw, hopefully you saw an email that I sent telling you about a poll, which is, I want to share with you. That's my email. Okay, so this poll here, I don't know how many people have actually done this yet. Let me click into it and see if I Zoom recording. Okay, so we're recording. I probably should pause it because we've got about 15 minutes to kill here. So I'm going to pause it. We're recording. Okay. So, all you people think you're so damn smart. Let's see if you are. Just pulling the quiz up myself a different way so I can read it to you. Okay. Pregunta uno. Okay, CRISPR-Cas9. Everyone loves CRISPR-Cas9, but does everyone know CRISPR-Cas9? I don't know. Let's find out. CRISPR-Cas9. What does the word CRISPR stand for in CRISPR-Cas9? I'm going to pick someone if no one puts their hand up. Someone I don't like. Who don't I like? Oh, I know who I don't like. Where are you? God. I like everybody in this class. Okay, well, let's just pick somebody. Esha Gupta, what does CRISPR stand for?
A clustered, regularly spaced short tandem repeats.
Okay, so now in the actual experiment, what is the CRISPR part of the experiment?
Doesn't it like guide the Cas9 to the target sequence of the gene
Right,
that they're trying to?
and how does it do that? What is it?
That I do not know.
Okay, anybody know what chemical the CRISPR is? How about Hannah Rondo?
I'm honestly not sure.
No? Okay. Isun, you seem to know everything. What is the CRISPR?
No, I also don't know this one.
Oh, come on, who knows it? Mairead knows everything. She knows a lot of things anyway.
I thought it might be an RNA sequence.
Yep. Anything else you can tell me about that RNA sequence? It's kind of important. So the CRISPR is how you make this specific to one particular gene and not just inserting anywhere. So the CRISPR aspect of it is a little bit of palindromic sequence, and palindromic like reads the same way forward as backwards. So it folds up into a hairpin loop, and that sticks to the Cas9. And then there's more sequence coming off of it, and that's going to be complementary to the gene you want, the place in the genome you want to cut. So that's going to guide the Cas9 down to one exact sequence in the genome. So what's the Cas9 do then at that point? Michelle Simmons? I'm busting the women today. Next person is going to be male.
So what does the Cas9 do?
What does Cas9 do?
Cas9 helps target the gene.
Nope. That's what the CRISPR does, targeting. Could you repeat that about the CRISPR, please? Okay, so the CRISPR, hang on a second. Let's pull that up since there are CRISPR Cas9, send me a dream, bum, 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 bum. Make it the cutest dream that you've ever seen.
That Young Gravy.
What's that?
What song was that?
It's Mr. Sandman, Bring Me a Dream. It's a 50s era song, I think, but there was that a cappella science where he did CRISPR Cas9 using that song. So anyway, here is your CRISPR. Here's the little palindromic segment that forms this hairpin loop. And then here's the part that directs it to the right gene. And now this is Cas9. So that's what CRISPR is. It's this part of it. It's the sgRNA that's directing it. Now, what does Cas9 do once it finds the gene? Isn't Cas9 an enzyme? Yes, it is an enzyme. Thank you, Abdou Sabor. Doesn't Cas9 remove foreign sequences? Nope. Nope, it does not.
Does the Cas9 got the DNA at that specific location that you're looking for?
Exactly. Who was that? Who said that?
That was me, Abdul Sabor.
Abdou Sabor. Well done, buddy. So yeah, it directs it to the sequence, and then Cas9 cuts it right at that place in the thing. Okay. So all it's really doing is opening the genome up, and then things you do afterward, either close it back up there, which is going to just because everything is imperfect, you're going to lose some bases there and screw that gene up. So you're knocking the gene out. Or once the Cas9 has opened it up, you insert your own gene in there, and now you've got a different gene in the exact same spot as that old gene. It's magic. It's magic, I tell you. Okay. Next question. Stop share. Question 2. Oh, I like this question. Why are some important signal transduction proteins designated with names like disheveled, shriveled, and jagged? Who's got this? I talk about this a lot. And it's not that important, but it does bring up something that we talked about in class Friday.
Doesn't it
Hannah?
have to do like its name for the phenotype?
Of what? Yes. Yes, you're right. Phenotype of what though?
Of the organism, like mutation?
Yes. Do you remember what the organism was?
No, I don't remember.
Okay. Drosophila. So one of the things we talked about was forward genetics. Or was it reverse genetics? I think it was reverse genetics. Let me pull that up real quick. CRISPR-Cas9, send me a dream. And no, I don't think I had a thing on that. Well, anyway, I'll pull my notes up. Okay, reverse genetics. I always mix the two up. Oh, no. Forward genetics. Forward genetics. Sorry, guys. I should know that. So forward genetics, the idea is you take a whole bunch of like eggs or early embryos and you bombard them with UV or X-rays or something that's going to mutate genes, and then you see what comes out of it. And you name the phenotype for what it looks like Hannah said. So that's forward genetics. And that's why they're named weird, because they're named for the phenotype that resulted when a specific gene got knocked out. Okay, last question. Before the advent of technologies like CRISPR-Cas9, insertion of a new gene into the genome of another cell, I should say cell, not cells, into the genome of another cell was done by random insertion, non-homologous recombination. Lists 3 reasons why CRISPR-Cas9 is far better. Okay, why is it better to put a gene right where you want it versus randomly inserting? Hey, Tyler, I'm recording this, so you get a chance to look at the recording later. I want to keep moving forward because I'm running out of time here.
What was the question you asked?
So the question was, why is site-specific changes like with Casper, so you're going right into a specific point in the genome, why is that better than just inserting your DNA and letting it randomly insert into the genome?
It can guarantee that the inserted gene is in a transcribed region.
Or no, what region? I'm sorry?
Like a transcribed region, like it will be, it's not in like exons and stuff.
Right. Yeah, there are whole regions of chromosomes that are silenced. And if you put your DNA in there, you'll never see it. You'll never know it. So you make sure you put it in a region where it is going to be expressed. What else?
Mutations.
Mutations. Okay. What do you mean? Say more about that, Yusuf.
Would it be like there's different types of mutations? So would it be like missense or like silent mutations or like I don't know?
Well, you're defining different types of mutations. The problem with random insertion is you can get mutations, but why is that a bad thing?
I honestly don't know.
Okay. Well, if you're just inserting randomly, you could knock out a good gene, right? And then you lose function in that good gene. And then the phenotype you get after that, is it because the new gene you put in or the old one you knocked out? It gets kind of confusing. And so if you can target, if you're changing a gene, you want to go from one form of the gene to another form of the gene, you can go in and at the same time knock out the old gene and put your new gene in there. It's a much cleaner experiment. And also you can knock genes out easily, which is a different advantage here. So in the old way, if you wanted to knock out a gene, you really couldn't because you couldn't target it. You would have to do something like randomly insert a gene that would give you the complementary sequence to the gene you want to knock out, and then that messenger RNA would knock out this messenger RNA. Tricks like that, but being able to target the exact gene is just a lot more efficient.
Doesn't that allow you to be able to edit multiple sites at a time as well?
That's true. That's another thing you can do with CRISPR. You can change 2 or 3 genes at the same time, which can be an advantage as well.
What was the first reason?
Mairead said you can be sure you're putting the DNA in an active region. So one of the ways that genes are controlled is by either condensing or expanding the genome, the whole genome in that area. So if your gene ends up in an area of a chromosome that's all condensed up, it's not going to be expressed. So you want to make sure you put it in a place where it's going to have the opportunity to be transcribed. That makes sense, Jason?
Yes.
Okay, good. All right, 1:33, we've got 15 more minutes. Let's go ahead and get started on chapter 4 because I like chapter 4. Close. Okay. Share screen. This is not what I want. Let's close this. But okay. How much of our class time is taken up by me going to different folders? It's annoying. Okay. Xenopus laevis. The Xenopie. I just made that word up. That's not a real word. Just getting my stuff together here. My notes over on the side over here so I can follow along. I should put these right in the PowerPoint. That would be annoying. Okay. So here is your life cycle. We talked about this in the last chapter. And in the last chapter, basically we were looking at the what happens. You know, how are the tissues moving or whatever to get you your 3 axes and your adult organism. Here we're going to be looking at why. What's going on in the cells that's causing all this stuff to happen. So back in the last chapter, we talked about gastrulation in the frog, where the cells divide and divide and divide, and then you get this dorsal lip over here, which we call the organizer, and then endoderm and mesoderm cells come in this way and crawl across, and that's what gives you your gastrula, and then that neurulates. So that's what we talked about in the last chapter. So now why? Why is all this happening? Why is this happening to me? So another aspect of this is the whole idea of fate mapping. Like what's going to be made of this part of the early embryo in the end. And you can just, by looking at the embryo, if you know where the sperm went in, you can tell exactly what each part of that embryo is going to be in the adult. Excuse me. I missed a chat there. Chat. Marie, can you read the last chat message? I can't pull it up here.
I just had a question about the CRISPR-Cas9, but I can wait till later.
Okay, I'll remember to hang out after class and then we can talk. I cut you off last time.
Yeah.
Okay, so, okay, so anyway, whenever you see a picture like this, basically we're assuming the sperm came in this way. Sperm can only come in the animal pole. It can come in anywhere in the animal pole. But if it comes in here, what happens is the tissue over here that was endoderm, or no, it was ectoderm, becomes mesoderm, and it will end up being the notochord. Everything in the middle is mesoderm, but you've got ventral mesoderm versus your middle mesoderm, your somites, and then your notochord over here. And, you know, at this stage, it's not that, but this is what it's going to be. That's why they call it a fate map. When this gastrulates and then neurulates, this section ends up all over here. The endoderm still stays on the bottom. The epidermis covers the bottom, so you can see that's coming down here. So originally those cells are up here, and they end up on the ventral side of the organism. So that's what fate mapping is. So the fact that at the early embryo, you can tell where all these tissues are going to be tells you that there must be something in the egg that's causing this to happen, because it's not like determined by the sperm coming in. The egg is sitting there ready to go, and this is what's going to happen no matter what. So, and again, the only thing that matters in terms of the sperm is where actually that sperm comes in. Okay, so here's our embryo. And the reason we can fate map is because here, oh, this is an embryo. This is just the egg. You've got an animal pole and a vegetal pole. You've got the yolk on the inside, and then at this point, it's just one big cell. But inside this egg, you've got different mRNAs at the top versus at the bottom. So these are maternally put in there. They're put in during oogenesis, and they're restricted so they can't diffuse around. And they're also not being transcribed. So you've got a whole bunch of messenger RNAs that are just sitting here and here, and that's going to do your fate map in the end, do your determination of what tissues become what. The big event of the nerve or the sperm fusing then is going to tell you that will be the ventral side. Wherever the sperm comes in will be ventral. Directly opposite will be dorsal, and that's because of factors that we'll talk about later in this chapter. Okay, so in one way you can tell that even the early embryo is fated in certain ways is by what happens when you separate cells during the early cleavages. So at the very first cleavage goes vertically like this, and it usually goes right down the plane where the sperm came in. So that's where your first cleavage is. Now, if you take these apart, split them apart so you've got two cells, they both become perfect embryos. Maybe a little bit smaller than a regular frog, but they'll develop into full adults. And that tells you that each of these cells is totipotent, totally potent. It can become anything. So at this 3-cell stage or 2-cell stage, these cells are identical to each other. Now, when you get one more cleavage, another vertical cleavage this way, now these cells are not the same. Okay, this is the plane that the sperm came in on. Remember I told you that if the sperm came over here, this is going to be dorsal. And that's already been set up at this point. Even though it's only 4 cells and there is no dorsal, there is no ventral, this will be dorsal. And if you split these cells apart, so you've got these 2 cells over here and these 2 cells over there, what happens is these 2 become most of an embryo, but they end up without any gut. So they really don't get any endoderm. Even though you can see a lot of endoderm here, that ends up turning into mesoderm. So if you take these 2 off, you've got a dorsalized embryo. It's a lot of the dorsal structures, but none of the ventral structures. And these over here lose all dorsal and anterior structures. So they end up being just ventralized stuff. They call that a ventralized embryo. So basically it's just a ball of gut, okay? So At this point, we call these cells pluripotent. They're not totipotent anymore because each one can become many different things, but not everything. Okay, at this point, there is some specification that can't be changed. Ventralized, okay. And then, of course, you do your third cleavage. It comes across the equator like this. And now you've definitely got two different tissues here. In the last slide, this cell has got ectoderm and endoderm on it. This has got ectoderm and endoderm on it. The next slide here, this is all ectoderm. These will be more dorsal ectoderm. These will be more ventral ectoderm. This is going to be more dorsal endoderm, and this is more ventral. But now you've got, again, they're pluripotent. They can become many, many things, but now they're definitely specified to move in a certain direction. They're no longer totipotent. So that's mainly what we're talking about in this chapter is like, what are the molecular factors that cause this to happen, that specify tissues to become different things? Because I mean, you could imagine taking one of these cells out and just developing into an entire embryo, but it's too late already for this guy because of the maternally laid down messenger RNAs. So here's that experiment where you slice across at the 4-cell stage. So you've got these dorsal determinants here, right? The sperm came in here and that put dorsal determinants off of it. So when you split them up in half, here's your dorsalized embryo, no gut. Here's your ventralized embryo, just a ball of gut and nothing else. And here's your normal embryo. So you can see a difference between the dorsalized and the normal. Okay, so what is it that's going on? All right, so we've got a number of messenger RNAs here that are going to be attached to the cytoskeleton. And basically they're attached to the cortex. Let me see, just cortex next, yes. I'll come back to this slide, but if you had cell biology with me, I talked about this in the first couple of lectures. The cortex is this layer of cytoskeleton that's right under the plasma membrane. And here it is here in a drawing, okay? And so that's holding up the plasma membrane. We've got these structures that go through the plasma membrane and attach to this. And this is kind of like hands holding the whole plasma membrane out. And it's like a mesh, like a gel, excuse me, surrounds the whole cell. Now, these are not round cells. They're spherical, I mean. This has got a cortical mesh all around it in 3 dimensions. And it's just this thin layer on the outside next to the plasma membrane. So in the egg, the Xenobus egg, we've got a number of messenger RNAs that are attached to this cortex, and they're in the vegetal pole, basically, right most extreme bottom of the vegetal pole. And these are VEG1. VEG1 is also called GDF1, although I've never heard it called that. Your book says it can be called that too, so we'll leave it there. But mainly we'll be calling it VG1. So VEG1 is a TGF beta-like hormone. We'll talk about the TGF beta pathway shortly, but it basically is just a signal transduction pathway. And there's a whole family of hormones that work through this pathway, and VG1 is one of them. Another thing down here is VEGT, and it's a transcription factor. And that's kind of how you remember VEGT, vegetal pole T, transcription factor. So here's a little shortcut I always use, just so you know. TX means transcription. So a transcription factor is a protein that will bind to the promoter region of a gene and then turn it on. We talked about this a little bit on Friday when we were talking about cross-linking proteins to their promoter. So this is a protein that will bind to a promoter and turns a specific gene on. Beta-catenin, another transcription factor. This one is really key. It's a very important transcription factor. We see this a lot in this class. Wnt, this is Wnt11. There are a number of Wnts. Wnt comes from the phenotype wingless from Drosophila. Wingless and int, 2 different proteins that are the same, so they called it Wnt. But anyway, Wnt 11 is a growth factor, so it's kind of like VEG1. It's a hormone and it can diffuse away from the area that it's made. And disheveled then is a signal transduction molecule. So it's on the pathway of Wnt signal transduction. So we will talk about Wnt signal transduction, and then disheveled will make more sense when we do that. But basically, these are the vegetally restricted factors. These are going to be causing the vegetal pole to be endoderm, and these are going to be the dorsalizing factors. These three things are what are going to cause one particular part of the endoderm to become an organizing center that can cause dorsalization. So right here at the very beginning, you've got beta-catenin all over the vegetal pole. You've got veg-T here, and you've got Wnt and disheveled here. They're all attached to the, or messenger RNAs for them are attached to the cortex. And then Mr. Sperm comes along and comes in at one site in the animal pole, anywhere in the animal pole, but only in the animal pole. So when it comes in here, what happens is this thing called cortical rotation. So the whole cortex now is going to rotate in this direction. So it rotates so that those dorsalizing factors that were attached to the pole of the vegetal side roll up the side and end up over here. Now that's going to be your dorsal specifying tissue right there. That's what's going to specify dorsalness there. And you can prove this. The way they proved this was they hit eggs with UV light, and that disrupts this cortical rotation. What happens is you don't get an embryo, okay, because you don't have a dorsal side, so you don't get a dorsal ventral axis, you don't get anterior posterior, you don't have anything, it just becomes a blob. And you can take these the ventralized embryos that you've done this UV trick with and just turn them a little bit. And when you turn them, that cortex will rotate, and then boom, you rescue the dorsalized embryo or the ventralized embryo. So that's your cortex and your cortical rotation. So cortical rotation puts those things there. And so now you've got disheveled, Wnt, beta-catenin, and another protein I didn't say before, GSK3B, which is GSK3 binding protein, which will bind up this protein GSK3. These are all going to lead to an increase in beta-catenin in these cells and not other cells, okay? And then to make it a little more distinct, we got another protein we'll talk about later in this chapter called Dickkopf. Anyone know what cough is in German? Dummkopf. Cough means head. So you can tell that the embryologist who came up with this name had a good sense of humor or a bad sense of humor. Dickkopf, DKK1. And this blocks Wnt signaling, and it's located in the ventral side and it's diffusible. So that's a theme we'll see a lot in embryo development, where you've got one specific area making a diffusible factor and it diffuses away, and then you get a gradient of effect based on the concentration of that thing. So we'll see that a lot in this chapter. So anyway, bit of an introduction to cortical rotation and the development of the dorsal ventral axis. We'll talk a lot more about that in this chapter. Okay, any questions? That's about as far as we go. So I'll send you an email about the results of that poll. If you could just please do your vote like in the next 15 minutes, because I'd like to wrap that up so we can figure out what we're going to do this week. Any questions?
Dr. Slush? Yeah, this is not content related, but I would just like to point out that today is my boy Stefan's birthday.
Oh, really?
Yeah.
How old is he?
Shout out.
How old is he?
Stefan, how old are you?
No, not you. I don't care about you. Your son, how old is he? Yeah. Okay. Okay. All right then. See you guys later. I'll stick around, talk to Mairead.